Welcome everyone back to Behind Our Science, where we uncover the stories and people behind cutting-edge science. In light of recent policy changes, shifting away from animal research, a lot of the different outcomes from NIH changes, we wanted to learn more about alternative research approaches and how people in biomedical research, including doing cell work, what we refer to as in vitro, or doing glass dish, or petri dish, many different types of in vitro work, and also organ on a chip, which is a thing that we're gonna go over. And what does it really mean to have an organ on a chip?
So thank you for joining us at Behind Our Science. We're here today with Dr. Lans Taylor from the University of Pittsburgh, which I'm at now too. He's going to share about his career, his work on human microfluidic systems, and possibly teach us a little bit about fly fishing if we're lucky. Dr. Lans Taylor is a leading expert in live cell imaging and human microfluidic systems to study complex diseases. Dr. Taylor is a true innovator with 29 patents, multiple startups, and over 300 publications. His work spans microfluidics into cellular physiology and crosstalk, allowing his team to screen drug responses to treatments in a high throughput manner. Dr. Taylor has worked in industry and academia and has great perspective on what is yet to come in the world of biomedical research.
Welcome, Dr. Taylor.
Hello, it's a pleasure to be here.
Thank you very much for taking the time to talk to us today. So our first question is, what is a microphysiology system?
Well, microphysiology systems, or MPS as they've come to be known, are miniaturized functional units of organs constructed usually with multiple types of cells that are the key cells within a given organ, and under a variety of both physical and biochemical environmental cues, such as electrical stimulation, flow stimulation, or mechanical stimulation that recapitulate at least some organ or organ systems physiology and pathophysiology. And there's two types, which has been a bit confusing in people coming into the field. One we've called structured biomimetic MPS, and these are different organ cells that are layered or bioprinted to recapitulate the structure of the organ, like the liver, a sinus in the liver, and it can use primary cells or induced pluripotent stem cell derived cells. And the second are organoids, and organoids are three-dimensional structures derived from pluripotent stem cells, progenitor and or differentiated cells that can self-assemble through cell-cell and cell matrix interactions to recapitulate aspects of the native tissue architecture and function. So these two types are the main drivers within the field today.
I mean, a lot of this information might be new to our listeners, but I mean, in an optimization of these systems takes a really long time. And so one of the other follow-up questions that I had for you is there a limit of how many cell types or the total cell that you can utilize in these systems?
No, there's no limit. In theory, you can actually build a whole organ, and there are people trying to build livers, for example, that can be transplanted so that people at some point in time can avoid a transplant and actually receive a whole, quote, new liver. This is something that has some limits in terms of the engineering, in terms of the size of the system, et cetera. So these MPS can scale from something the size of a slide, a microscope slide, up to a small box if you're generating kind of a whole organ, or at least part of an organ.
Sounds good. I mean, there's a lot of approaches into the organ on a chip and other systems trying to build organs completely. So that's really amazing. So talking about the MPS, the microphysiology systems, like liver on a chip. I guess the question is, are these optimized to model metabolic diseases? I mean, these are complex. Metabolic diseases are complex. And as such, is there ways that to improve reproducibility across patient populations?
Well, I think there's two questions here that I'll address.
Yeah.
One is demonstrating reproducibility between MPS that are constructed from the same materials with the same protocols. In other words, can you reproduce the same experiment using the same materials and approaches time after time? That's critical because the real value of these MPSs are defining, quantifying the differences between patients. So you first have to set the boundary in terms of how reproducible are the systems when you use the same exact cells, media, matrix, etc. And then you can quantify differences between different patients. And in fact, what is becoming now a dominant approach is building MPS from patient cohorts, where you first generate induced pluripotent stem cells and then differentiate them into the distinct cell types within the organ. And these models we call patient biomimetic twins, because it literally is building a liver, a lung, or a kidney model from the patient's own cells. And in parallel, major advances have been made now of building what is called patient digital twins, and that's taking data from those patients where you've derived the induced pluripotent stem cells, and you put together all of the clonomic and a lot of multi-omic data from each patient, and we call these patient digital twins. They can be used to make significant predictions about the patient, whether the patient will exhibit toxicity to a particular drug or whether a particular drug will have efficacy in that patient. And you can test it in the patient biomimetic twin before you actually apply the drug, for example, to the actual patient.
Yeah, that's amazing. I guess that also helps with prognosis, I guess, in some way.
Absolutely.
And so these MPSs, again, from your perspective, for diseases that have either chronic, slow evolution, development, rare diseases, for example, cholestatic liver disease, can these be applied to those?
Yes, and in fact, one of the main areas we've been studying the last number of years is in liver disease, which used to be called non-alcoholic fatty liver disease, which is now called metabolic dysfunction associated liver disease.
Yeah, the MASL thing is now a whole thing.
And it essentially emphasizes the challenge that we have in developing therapeutics for patients with these complex heterogeneous diseases, because everybody's different, and that's been shown. In the last decade of trying to develop drugs for MASLD, there have been mostly failures because the companies have failed to meet endpoints, which in a poor way has been defined as just a percentage of patients that respond. And in fact, what is needed is a technique, and we can use these patient digital twins and patient biomimetic twins to identify subcohorts that might be more responsive to a particular drug or show more toxicity to a certain patient subpopulation. So we are on the right track. It's more complicated than the pharmaceutical industry would like. They'd like one drug to fit all because that's the most efficient, but the patients decide what they will respond to.
Yeah, and it's very, very heterogeneous. So I guess you already answered my next question a little bit, but I guess in that context of screening data for integration of quantitative systems like pharmacology to predict drug efficacy, you also mentioned toxicity. Are we there yet? Is there something that's being done in that sort of scenario?
Well, I think if we look back, which is pretty amazing, the whole human microphysiology system technology started in earnest about 15 years ago with funding from the Defense Department, DARPA, and early funding from NCATS at the NIH. And within 15 years, we've gone from most people believing it couldn't be done, including the then head of the NCATS, to now it's accelerated to the point where, as you pointed out, their NIH is kind of downplaying the use of animal models in disease, and the FDA is accepting these microphysiology systems and computational models without requiring, as it has been in the past, animal data. So I think in 15 years we've come a long way. There's still some mountains to climb. I think the slope is steep and people are excited about the potential, but my own projection is within 5 years we will be able to do some very significant things.
Thank you.
Amazing. Do you think that there are some computational strategies that best capture cellular and tissue level response variability in drug testing now, or is that something that maybe we're going to grow into?
Well, there's been some things that have been done actually going back a number of years using so-called quantitative systems pharmacology, which is a method where you have an iterative use of experimental models and computational models. The experimental models can be used to test predictions from the computational models, and then the results can be used to update the computational model. So people have done things, and we've done some of this, doing repurposing of drugs by using RNA-seq and then looking at differentially expressed genes and enriched pathways, and then predicting drugs from drug bank that have a chance of working on a particular subset of patients within a cohort. So that has done a great deal, but it's also led to the next level. And that next level is what I described as patient digital twins. This is taking and literally making a computational model of a patient from all of their deep clinomics data, as well as genomics, proteomics, metabolomics, etc., and then building a model where you can ask questions about drugs or toxicity. And then again, the next step of MPS are these patient biomimetic twins, where they are cells derived from the same patients that you've made the patient digital twins. So I think there's an impact that's occurred in the recent past, and now the future, at least in my view, is this integration of patient digital twins and patient biomimetic twins.
Amazing. And do you think that these biomimetic and digital twins are what helps making human MPS qualified and validated for regulatory approval?
Yes, and in fact, we were fortunate enough within the past year of receiving funding from the NCATS, working with FDA. These are called the translational centers for MPS. We're one of just a small number, and we're working with the FDA to qualify some of our MPS for their use. And for qualification, all that means is that the FDA can accept at face value the data from a qualified MPS, just like they have qualified animal models in the past.
That's so cool. I love it. I wish I could do that. I don't know. I still work with animal models, so this seems a little over my head, but it's amazing. It's amazing.
Well, I'll just make an aside comment. I think that the NIH is being a little premature in shifting fully away. I think there's still value in animal models, and I think a spillover time where people are doing things both in MPS and in animal models might be telling. But I think the future clearly is these in vitro models, because they will become more and more sophisticated, including getting the point where we're producing a liver that can be transplanted, or even a kidney, or etc. So that's the future, but there's still value in some animal studies.
I mean, for my job's sake, I agree. But also, like, I study cholestatic liver diseases, and the liver transplant list is hard to get on in the first place. And then if you're diagnosed late and you start having these comorbidities, it just makes it harder. So I think you're right. These in vitro generated organs are indeed the future. To shift a little bit to fluorescent imaging, what roles do fluorescent reagent technologies and biosensors play in advancing real-time and dynamic phenotyping of 3D tissue models?
Well, in one word, it's huge. But this was one of my early fields that I've kind of continued tapping into with the MPS. So with fluorescence-based biosensors, you can engineer cells to be sensors of different activities, like calcium activity, oxidative stress, the generation of fibrosis that can be quantified in real time, and with labeling different immune cells, monitoring their migration from the vascular channel in an MPS into the actual organ tissue level. So they're very powerful. There's also the electronic so-called inline sensors that can use different detection tools like antibodies to induce electronic signal changes for various excreted factors, glucose and various hormones, etc. So I think these MPS are going to become even more sophisticated as we're monitoring in real time a variety of key pathophysiological and physiological activities.
No, very true. So speaking of what your research you used to do that you're still tapping into with these MPS systems, and knowing that you have been in both academia and industry, how did transitioning between these two sort of overlapping but still very separate research fields shape your leadership skills in increasing innovation and team building?
Yeah, that's a great question. I made the transition from academia to industry. My wife pointed out to me that I had just turned 50 and had a midlife crisis. And instead of a sports car, I left Carnegie Mellon University and started a company. But so I had no idea what I was up against. But it was clear to me that when we decided to build a whole platform for high content screening, which meant instrumentation, software, and reagents, that this would take much longer in academia than creating a company and going at it full speed. So how I was shaped having that experience was understanding the rate of starting and completing projects is critical. It's more optimal not to use the meandering approach of academia, because it can work that way, just going from one grant to the next, but having a target and then going after it full speed. Team science brings together expertise to move faster and smarter for common goals. Mm-hmm. And that's both in academia and industry.
Yeah.
It requires that the team science, that the team members, especially in academia, get clear credit for what they did in the project. Because in industry, it's the whole team, and it doesn't matter.
Yeah.
Somebody's boss is going to determine how valuable somebody is. And in academia, it's publications and giving talks, and you have to highlight team members while you're also integrating the ultimate team effort.
Yeah, no, that's very true. It feels like publications are currency in academia.
Yeah.
Yeah. So based on this experience, like, what skills would you say are absolutely essential to lead these multidisciplinary teams?
Well, what I learned through the School of Hard Knocks was that you really need to understand the complete technical components and how they fit together in one of these interdisciplinary programs. To lead it, you don't have to be an expert in each element, but you have to know enough to see how it fits together to be able to then go out and pick the best collaborative people to accomplish the goals, and then drive the communication and language needed between these disparate disciplines. And then I think most importantly, giving credit to the whole team and individual members when they're successful.
Amazing. Love it. Thank you so much.
Yeah, that's great advice, Dr. Taylor. And I have to say, like, a lot of the things that you've been saying, a lot of our listeners are either from academia, also from industry. As someone that recently transitioned from academia and is starting its own company, I think these are fantastic points to actually be able to successfully do so. As such, I was going to ask you a couple more questions, particularly about this. But you've been successful doing both things. How does one successfully commercialize academic inventions, things that you've been working on for multiple years, with the things that you mentioned about the ventures from academia and the slowness about academia, and now into industry, and transition them into sustainable biotech ventures?
Well, again, this isn't something, any wisdom that I was born with. The School of Hard Knocks, as I often tell my colleagues, as well as my children when they were younger, School of Hard Knocks is the best teacher. So yeah. One of the things that I learned is that if you have a good idea, and it's something that could be transformational, you need to patent it. So you have to take the time and essentially work with a good patent attorney to draft a good, at least to start with, provisional patent so you can protect your idea for its potential value commercially. I would also say you can take advantage, if you're in academia, of organizations like we have at Pitt, the Innovation Institute, yeah. But you also want to do your own market research. And I'm not talking about doing surveys, etc., but actually determining as a practitioner in a field, is there or should there be a demand for the invention that you're working on? Because if you just do something cool and nobody cares, then it's an academic activity that could be valuable academically, but not necessarily commercially.
Sure.
Another point is that not all, quote, money is equal. And from experience, there are excellent investors that bring value beyond their money. And there's also bad investors. And I've had the joy of dealing with both. Mm-hmm. And then the final thing I'd say, and this is really important, obvious but critically important, hire the best technical and business people that can interact with others to accomplish tasks.
That's great advice. Thank you very much for that. I mean, as people that are doing, I mean, we have people in different stages of their career. So we get from very early on to like fully blown academic professors or people that are already in faculty positions. Are there strategies that help either senior researchers or people that are getting into that stage of their careers at the cutting edge of technology, meaning like moving from, fluorescent microscopy to maybe AI-driven systems pathology? Are there strategies that you found successful for this?
Yes, and I learned this actually in a little bit in graduate school, and then made it a driver for myself. And it's simple-minded and obvious, but it's not always practiced. And that is look ahead to see what is needed in a field for significant progress. If you're in an existing field, do you keep doing the same thing over and over and over again, going to the 4th, 5th, and 6th decimal point? Or is there an opportunity to make a big step forward? And I would say that as an example with the MPS, animal models have had amazing impact for many, many years. Mm-hmm. And just 15 years ago, these little characters came along talking about MPS and keeping an eye on what was happening. It was an important thing, at least for us. And the other thing is, don't stay in. I'm speaking of myself now, and other people can deal with their careers the way they see fit. But I didn't like staying too long in the present. I wanted to help develop a field, develop it to a point, and then tackle the next challenge. And I think, to quote the known philosopher Wayne Gretzky from hockey fame, you want to see where the puck is going, not where it's been. And so in that regard, I like to encourage people to follow your own nose and not the herd.
That's great advice.
And that's risky because
it is very risky.
A system, but again, you have to decide, why are you doing this, whether it's in academia or industry. And I hope if most people, it's to really understand something to the point where it can have a positive impact on the world. And that isn't always doing what others are doing. And so I'd rather be risky and fail, which I did sometimes, than to be too safe.
Yeah, and I think we mostly learn from our downs than from our gains. So
no question.
Yeah, so that's great advice. The last one before we move on to the other side of our non-scientific questions is, what advice do you have for young scientists aiming to combine, again, wet lab to dry lab discovery, computational modeling, or even translational impact? Because this is an area of people where a lot of the NIH changes are shifting. Like I said, for me, it's made and resonated because I'm in that transition. And I guess a lot of people are also in that same venue where they're understanding, I've done my work, but I also want to combine this with other aspects that I really want to take on. So is there something that you would be able to advise them as you've been successful in your career?
Yeah, and again, it starts with taking a step back. If you're interested in a particular field, take a look at where the field has started, let's say 20 or 30 years ago, where it is today, and where does the field, based on various reviews from the big shots, say that it's going.
Yeah.
And kind of think about the future, not the present. So, for example, if I were a young scientist today, I would want to do a thesis or a postdoc integrating in as serious a way as possible wet lab work with computational approaches. Because I think research in the biomedical clinical sciences anymore will use computational methods heavily. And we will continue to have wet lab, because we have to ask questions and then make predictions and then test those predictions. So that's what I would do. And you don't have to be a leading expert in all of those things, but having experience and a deep understanding of computational methods as well as experimental wet lab methods gives you a strong position, especially to collaborate with others to do even bigger things.
Thank you very much, Dr. Taylor.
Yeah, that's amazing. I definitely have been trying to learn to code for many, many years, and it is really hard because I'm a wet bench scientist, but I agree.
Let me emphasize that I haven't done any coding since graduate school. I didn't enjoy it, and I wasn't all that good at it.
Right.
But I understood enough about computer science and that I know how to talk to coders. Mm-hmm. And challenge, what's being done and how and why. So you don't have to do all of the details on all of the things you do. You need a good background in it.
Yeah, no, that's very true. I'll change my approach to it. So I have the joy of asking all the fun get to know you better questions. So one of my favorite questions to ask is, what is your at-bat song? Or it's also called a walk-up song. Like if you're on a baseball team, you're about to go bat, what would be playing?
Let's see, I think I'm a very optimistic person, because in life, I think you need to be, or you get depressed because the world's complex.
Yeah.
And even in dark times, try to see the positive side of things. So that song that kind of champions that perspective, the songs like "We Are the Champions," makes a statement that we're going to do it.
Yeah, no, that's a great song. It's very uplifting. This was behind a paywall, but I read that you like to fly fish. So how did you come into this hobby and lifestyle?
Well, it's interesting. A couple of things. My main hobby for many years has been doing science. And that's the joy of whether it's in academia or industry. If you really enjoy what you're doing, it is more of a hobby than it is a job. And so I've been fortunate to have that perspective. But with the encouragement of my wife, who said, look, you need some hobbies outside of science. Back in the early 2000s, a fellow that I grew up with as a kid contacted me from Montana. I hadn't been in touch with him for 30 years, 20 or 30 years. And we used to fish together as kids growing up. And he invited me out to Montana. He said, you need to learn how to fly fish if you're going to call yourself a serious fisherman. So I went out there and I learned to fly fish, and it was amazing. There's a couple of things. First of all, fly fishing is a combination of a strategic process. You have to be able to, quote, read the water. You have to choose optimal fly types for the insect emergence season, and then the technical skill of putting that fly right where you want it, where you can either see a fish or you predict a fish will be waiting to strike. It's also a great outdoors hobby. I mean, whether you catch fish or not, walking the rivers of Montana or western or middle or eastern PA is beautiful.
Yeah, that sounds lovely. And I also see a lot of marriage between fly fishing strategy and how you approached your career in research.
There was probably some logic there.
Some computational analysis.
Right. Yeah. Systems biology, the river. So what was your aha moment in your career that changed the direction of your research? You did mention a sort of midlife crisis going to industry, but maybe there was something a little more like eureka.
Yeah, this, and it happened. I was lucky. I was in a great department with a great mentor and great co-graduate students that interacted a lot. So I was very fortunate. So in graduate school, at that time, I was focused on understanding mechanisms of cell movements. And this is back in the dark ages, before we even knew that actin and myosin and microtubules were in non-muscle cells.
Yeah.
And so I was studying the giant free-living amoeba, because you could biophysically manipulate it because of its size and do biochemistry on it. And so I just asked the question one day, well, for the cell to move, does it need a cell membrane?
Right.
So a simple experiment was say, well, let's remove the membrane and see if we can find the right media that would permit or induce motion. And in fact, we were able to recreate cytoplasmic streaming without a membrane. So it answered the question, but it was random. So the membrane wasn't needed to be involved in generating the forces, but it was needed to maybe direct it. So that taught me a lesson of thinking outside of the box.
Yeah.
And furthering this idea, don't necessarily do what others are doing or others think are important questions. Follow your own nose.
Yeah, no, I do love that. I say follow your gut, but I like follow your own nose, because it is right there in your vision. You see it every day. If your research style were a movie genre, like action, mystery, or sci-fi, what would it be?
That's easy. Drama is destructive in science.
I see.
Sci-fi, if you look too far down the road, nobody will fund you. So it has to be either action or mystery. And I like mystery because it's solving problems.
Yes, no, very, very apt. Yes, I would definitely agree. Mystery is a good way to assign your research style. And if you had to pick a career outside of science altogether, which I know you called it your hobby, and like your approach to fly fishing is very scientific, so I'm very curious to know what your career would be.
Well, it's not all that different in detail, but music. I did music growing up, even studied at the Peabody Conservatory in Maryland, and thought about music as a career, but science won out in the end because music combines analytical thinking as well as the qualitative side of it, the sound, the quality of the art. And that intrigued me. I'm fortunate, although I didn't go down that path, I'm glad I picked science. My grandson, who is also a Lance, is studying music in college, and he's a natural. So
amazing.
I can live vicariously watching and listening to him.
Heck yeah, I love that. That's amazing. Congratulations to your grandson. Yeah. That's great.
Yeah, it's been fun.
That's awesome. Was there ever a moment in your career when you almost gave it all up, and what pulled you back in?
Yeah, I would say no. I never really thought to give it up. I had lots of failures from simple concepts and experimental designs that just didn't work. I've had papers rejected, grants rejected, people saying what you're trying to do can't be done. And then getting funding to do things that are out on the edge a little bit adds to the stress. But no, I was very fortunate. I liked kind of doing what I was doing and just tried to get up and keep moving forward.
Yeah, I love that. I mean, willpower is a hard skill to develop. So I aspire, and I hope that all our listeners do too. And now, what is one lab experiment that sounded absolutely ridiculous at first, but actually worked?
Well, this is an easy one. This occurred when I was at Carnegie Mellon, and I was collaborating with a brilliant chemist physicist, Fred Lanny, and we were using the light microscope to study dynamics of living cells, primarily with fluorescence, but other methods. So we were combining optimal optics and adding image analysis kind of things. But one of the classical things about the light microscope was the so-called Abbe limit of resolution, which was kind of a fixed law that people have been kind of studying for 100 years or more. And we made a mistake in setting up an experiment where we were trying to do what is called total internal reflection, which is just exciting whatever is closest to the bottom of a labeled cell to isolate it from the rest of the cell. And we made a mistake, and we mismatched the refractive index of the media, and we reflected light off the top cover slip that then created in physics called a standing wave in the field. And we saw when we pressed on the stage that there were zones of fluorescence that moved up and down through the cell. And so we asked ourselves, this was late one night, could we build a microscope and do an experiment where we could actually improve the axial resolution to see in the cell better and with higher real resolution? And of course it went against all the known physics at the time about the light microscope. To make a long story short, it worked. And it was called fluorescence standing wave microscopy, which then ultimately led to all of the ultra resolution microscopes that were developed in the 90s and 2000s.
That's amazing. Thank you so much for sharing these stories. This is so fun. I am so impressed.
No, we appreciate very much all of the insights, Dr. Taylor. I mean, to our listeners, we've learned so much about MPS today, but also being able to learn about your ventures and how you've been successful doing that. I think that's something that is valuable because it's really hard to get mentorship from someone directly, but also while they're hearing this. During our past episodes, we've had multiple people that have done ventures outside of academia. We recently had Dr. Jersh Perry, who started the Journal of Alzheimer's just out of the blue. And then he became the most cited journal in neurobiology just because. And he's become a great mentor of mine. We've learned about people that have published like 4 or 5 Nature papers in a year. We've been able to successfully do that, but hearing about people in the transition or in like the opportunities coming to them just because, it's a great impact, particularly when things like changes in the NIH or NSF or other different funding opportunities would actually affect our progression or maybe our trajectory. So we really appreciate your time today, Dr. Taylor.
Sounds good. Well, I enjoyed talking with you.
Yeah, so for everyone listening, I mean, this has been a fantastic episode to actually be able to learn about creating a full organ on a chip from MPS, but also importantly because there are novel approaches to producing patient digital twins and biomimetic twins as the future, potentially getting full liver transplants, but also mimicking these in other systems and organs. So we thank everyone for listening today. So everyone from Beginner Science, we'll see you in the next episode, and see you soon.